In this video, we will discuss about N-Glycan processing in the endoplasmic reticulum. We have already discussed N-Glycan processing in our previous videos. However, in this video, we will discuss this thing in slightly more detail. So let us first quickly revise how this N-Glycan initiate within the endoplasmic reticulum. So this glycan precursor, that is glucose 3 mannose 9 and N-acetyl glucosamine 2 will bind to the protein that is newly translated or during the co-translation with the help of this OST at a specific site, that is uh, NXST. This means that first amino acid should of the sequence should be asparagine, next amino acid could be any amino acid except proline, and the third amino acid should be either serine or threonine. So at this sequence, this glycan structure, that is glucose 3 mannose 9 and acetylglucosamine 2 will bind, and at this moment the 3 glucose structure are present in the glycan, so it is not recognized by calnexin and calreticulum. So glucosidase 1 and 2 will trim off the 2 glucose structure, and this mono glucosylated structure will then be recognized by calnexin and calreticulum, and this calnexin calreticulum will help or assist the proteins to acquire their native structure by either providing them the isolated environment and helps the proteins to get their native structure with the by making a complex or interacting with a protein from a PDI family, that is protein disulfide isomerase family, such as ERP57. So after some time, the glucosidase 2 will trim off the remaining single glucose present in the glycan. So now it is no longer detected by calnexin and calreticulum, and then this structure will be recognized or will be analyzed by UGGT1 and or UGGT2 to check whether the protein has acquired the native structure or not. If the protein has not acquired the native structure, it will then recycle them to the calnexin and calreticulum cycle uh, by forming by adding additional glycan to it. And after certain cycles, several cycles, protein is not able to acquire the native structure, then they will be moved to the ERAD mechanism. And if the protein has acquired the native structure after some cycles, then they will move towards Golgi apparatus. So we will discuss in this video about how this sorting will happen. So this is the normal structure that glycan that is going on within the endoplasmic reticulum. Initially, this is the triglucosylated glycan, and then this one glucose is trimmed off by glucosidase 1, and then second glucose is trimmed off with the help of glucosidase 2. So now the glucose 1 mannose structure is there. So after that, the glucosidase 2 will trim off, and glucosidase 2 can trim off the single glucose structure, and it can also attach the uh, single glucose to the non-glucosylated glycan, as it is shown here with the both arrows. And even if after several cycles the protein is not able to acquire a native structure, then this manosidase will trim off this glucose on the B arm. This is middle arm is known as B arm. So this will lead to a condition where the glucose where the it will trim up mannose at the B arm, not the glucose. Yeah, it will trim up mannose at the B arm. This will lead to a situation where uh, this mannose at A arm and mannose at C arm are susceptible to phosphorylation that can lead them towards lysosome, or they can move towards ERIC, that is endoplasmic reticulum associated degradation, with the help of ERIC 53, and these things are shown here in details. So, and this arm, the bottom horizontal arm is known as A arm, the middle arm is B arm, and the top horizontal arm is C arm. So when this glucosidase 1 is trimmed off, the first glycan trimming of the glucose trimming of the not glycan, trimming of the first glucose from this glycan structure. It will move towards the folding. It will move towards this interaction with calnexin and calreticulum. It is not shown here completely that after this another step will happen, that is glucosidase 2 will trim off second glucose and then will move towards folding, and this transport and sorting will happen. Transport and sorting will happen with the help of B arm, with the trimming of this manhose. So the trimming of this manhose, it will lead to two conditions, as I have already mentioned earlier, that either manhose at A and C arm will get phosphorylated and then they will move or direct towards lysosome, or it will be recognized by, or the manose 8 structure that will form after trimming of this manose from B arm will be recognized by ERIC 53 and they will move towards uh, further and glycosylation in the Golgi body. And this thing might be related, whether it will get phosphorylated or move towards ERIC will be might be related whether the protein has acquired a native structure or not. The trimming of C-arm. Trimming of C-arm will occur when the protein, even after several cycles, not able to get the native structure. Then trimming of manus at C-arm occur. That will leads to a structure that is recognized by EDM1 or EDM3, and they are related to ER associated degradation, or we can say they will transport the protein towards ubiquitination. Proteasome degradation via ubiquitination. Okay. So this is how the protein sorting occurs within the endoplasmic reticulum, and that is related to the glycan structure. So I hope you have understood some of these things that has been discussed in this slide. If you need to understand these things more in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.